Time up. What? Good evening, friends. Let us continue with the topic that we just discussed. The differences between the DNA replication in prokaryotes and DNA replication in eukaryotes. So remember, the DNA replication is a process to duplicate the chromosomes. Chromosomal duplication only possible through the doubling of the DNA content. And whenever the DNA content go for doubling, there should be no change in the chemical nature. Just to take what the replica. And this process only we call it as what the DNA replication. The process of DNA replication we already discussed. Then let us see the major difference between the replication in prokaryotes DNA and eukaryotic DNA. So remember all the time this prokaryote, DNA is naked. It is not inside the nucleus. Whereas in case of eukaryote DNA, it is always inside the nucleus, only protected with a double layered membrane. We call it as what the nuclear envelope or karyotheca. So let us see the differences between the DNA replication in prokaryote and eukaryote. The first one, in prokaryotic cell, it occurs in the cytoplasm only because no nucleus. Their DNA is naked without any nuclear envelope or without any cover. Whereas in case of eukaryotic cell occurs inside the nucleus, the DNA is there in the chromosome and chromosome in the nucleus only. So naturally, this will take place in the nucleus itself. And only one origin of replication in case of what the prokaryote. It is having a circular DNA. You already discussed. DNA is circular. So only one place it's originate. Oocytes only one. And from there it start and move to what you can say another end and finally reach there. Only origin of replication. Whereas in case of eukaryote, there are many what you can say origin of replication or oocyte because it is linear. Many are there. So that it won't take much of the time to finish the process of replication in eukaryotes. And third one, origin of replication is about 100 to 200 or more nucleotide in length. The oocyte. And oocyte is nothing but the sequence of nitrogenous base only. In case of prokaryote, that oocyte consists of around 100 to 200 nucleotides only in that oocyte. Whereas in case of eukaryote, that oocyte consists of only around 150 nucleotides. Comparatively, it is smaller. And in case of what the prokaryote, replication occurs at the point in which chromosome. Replication occurs at one point in chromosome, each chromosomes. Or that is what only one oocyte. Whereas in case of eukaryote, replication occurs at several points simultaneously in each chromosome. Every chromosome, we can find number of oocyte to start the process of replication in case of what the eukaryotic cell. And because of what the only one oocyte, in case of prokaryote, only one replication fork, whereas in case of eukaryotic cell, multi-replication forks are formed simultaneously in each chromosomes. Then only have one origin of replication. Here, multiple origin of replication in case of eukaryotic cell. And initiation is carried out by the protein DNA A and DNA B. In case of what the prokaryote, initiation of this replication carried out by the protein DNA A and DNA B, whereas initiation is carried out by the origin recognition complex in case of the DNA replication in eukaryotes. And DNA gyrase is needed in case of what the replication of prokaryote, because DNA gyrase is what a type of what topoisomers only to reduce the tension during the unwinding of the DNA. Whereas gyrase enzyme is not at all needed in case of what replication of what the eukaryotic DNA. So these are the basic differences between the replication of the prokaryotic DNA in prokaryotic cell as well as what in the eukaryotic cell. Then we have to discuss about an experimental proof to explain that DNA replication is semi-conservative. Semi-conservative means during the process of replication, one DNA strand is conserved and another one is newly constructed. That is what we call it as what the semi-conservative. And we can prove this DNA replication is semi-conservative through some kinds of experiment. That is what the experimental proof for the DNA replication as a semi-conservative one. See, while replication, one strand of DNA is getting conserved and other strand gets synthesized by using old one as a template. Actually, template strand only conserved here, or the master strand only conserved during the replication, and another one is what newly synthesized by the DNA polymerase enzymes only from 5' direction to 3' direction. And it was first shown in E. coli and subsequently in higher organisms like a plant cell and animal cell, this experiment. And this experiment is done by the Matthew Michelson and Franklin Stahl in 1985. And this experiment asked many times in the board examination, Michelson and the Stahl experiment. They conducted experiment by using E. coli and a heavy isotope of nitrogen, that is what nitrogen-15, heavy isotope, not the radioactive isotope. It is heavy isotope. Normal one is N14. And here they use N15. It is what not radioactive isotope. Remember, it is a heavy isotope. It is heavier than ordinary nitrogen N14. And this experiment is called as what the Michelson and Stahl experiment. Michelson and Stahl experiment. To prove that the DNA replication happening in prokaryotes and eukaryotes are a semi-conservative one. Let us see the Michelson and Stahl experiment. Simple experiment only, concept is very simple. Here you can see what this experiment. First of all, they kept that E. coli in this N15 medium. Ammonium chloride medium only they've kept, and their nitrogen is what the heavy isotope only. NH4Cl, and nitrogen is what N15 for many generations, so that this heavy isotope of nitrogen incorporate both the strands of the DNA. Nitrogen is there because nitrogenous bases are there in this DNA, and this nitrogen present in the nitrogenous base is incorporated with the heavy isotope of nitrogen, that is what the nitrogen 50 only. After keeping this individual E. coli in ammonium chloride solution consists of what the heavy isotope of nitrogen for many generations. Then they have taken this E. coli after keeping in the heavy isotope of nitrogen for many generations and taken to what the normal ammonium chloride solution consists of what N14, normal light nitrogen only. And E. coli take only 20 minutes to multiply. As I told you earlier, before every cell division, naturally DNA content will multiply through the replication only. So after 20 minutes, it replicates, it multiplies its body. It takes only 20 minutes to multiply. And they analyzed that the DNA, after 20 minutes, they found that this DNA produced in the first generation having one heavy isotope and another one is what the normal nitrogen only N14. That means this heavy isotope, it is incorporated in the DNA, produce a new strand of DNA that is with the, what the nitrogen 14 only, and it is what the hybrid type. And here also it is hybrid type, and they kept this E. coli in the same medium consist of what the N14 for the next generation to come. And they got the next generation, and here what happened, this will produce two DNA, and here we can get, they got what a hybrid one, and here they got light strand. And same way here also they got one hybrid strand. One is heavy and another one is with a light nitrogen. And here they got the light one. Both the strands are light. Answer is very simple. While the what you can say replicating this DNA, both the strands are one strand is conserved here, and that's conserved strand only used for constructing what a new strand here, and another strand is conserved here, and that only used here as a template for making or synthesizing another one. That is why in the first generation they got the hybrid DNA with what you can say light nitrogen as well as the heavy nitrogen. And they go for the next generation, same thing. This DNA, at the time of replication, these two strands separated. That heavy strand produce what? Another light strand, because it is there in the light medium only. So by using this nitrogen 14, they constructed a new strand here in the second generation, so that it is a hybrid one. And the light strand already present in this first generation DNA act as a template and constructed another one. So both the strands are with light nitrogen only. The same thing happened here also. During the replication, these two strands separated and act as a template, and the heavy strand produce what? One light strand, and this light strand act as a template and produce what? Another strand. So here, replication is purely semi-conservative, because during this replication, one strand is conserved and another one is newly synthesized. And that conserved strand only we call it as what? The template or the master strand. And this is only giving the information regarding the construction of what? The new DNA strand by the DNA polymerase enzyme that always elongate, as I told you, in the direction 5' to 3' direction only. So this is what the experiment conducted by these two scientists, Michelson and Stachel experiment. Let us see what are the different steps in the, what you can say, this experiment. Step number 1. See, these scientists grow E. coli in a medium containing N ammonium chloride, and here it is what? N15. Nitrogen is heavy. That is what? The heavy isotope of nitrogen only for many generations. First, they allow the E. coli to grow many generation in ammonium chloride solution, and here nitrogen is what? Heavy nitrogen, that is N15. Not radioactive, it is what? The heavy isotope only. So the result was the N15 was incorporated into the newly synthesized, because it is keeping for many generations. Naturally, both DNA strands will be incorporated with what? This heavy nitrogen available in its living surrounding. And this heavy DNA molecule can be distinguished from the normal DNA by centrifugation in not calcium, cesium chloride density gradient centrifuge. Cesium chloride, this is what the solution used in the centrifugal method, and that is why we call it as what? The CSCl density gradient centrifugation. Cesium chloride, once it's asked in the board examination, the cesium chloride, not calcium chloride, is what? The cesium chloride density gradient centrifugation. So naturally, it is very heavy. Nitrogen 15 is heavy, heavier than what? The nitrogen 14. So this N15 is not a radioisotope of nitrogen. So only the heavy isotope. So it can be separated from normal N14 only based on its density. You know, N15 is more dense, having more density, or it is denser than what? The N14. So in this centrifugation, that is CCl density gradient centrifugation, it settled down so quickly. Right. So this is what the way to separate the N15 from the N14. These two chromosomes can be separated. This chromosome DNA having N50 can be settled so quickly in this centrifugation method, cesium chloride centrifugation method, whereas N14 take time because it is lighter. Then second step, a transfer. After growing this E. coli in this medium for many generations, and incorporated this N15 in both the DNA strand, taken this E. coli into a medium with normal nitrogen, ammonium chloride with what? Normal nitrogen, and took the sample at various definite time interval. What is the time interval? 20 minutes only, because E. coli take only 20 minutes to multiply. And every 20 minutes, what happened? It go for the DNA replication. And through DNA replication only, it is able to multiply the genetic material. Then only it will divide. So they have taken the samples of this E. coli from what you can say, normal ammonium chloride solution at a definite time interval, that is 20 minutes only. Time interval was 20 minutes. This is the time required for E. coli to multiply its DNA content. So various samples were separated independently on cesium chloride gradient centrifugation method and measured the density of the DNA, and result was analyzed scientifically, right? And third step, DNA was extracted from the culture one generation containing heavy isotope of what? The nitrogen N15 consists of heavy double helical structure of DNA. Both the strands of DNA made with the N15 only. See here, both the strands are made with the heavy isotope of nitrogen, because they kept this E. coli in this nitrogen, heavy nitrogen isotope for many generations. Naturally, that nitrogen, heavy nitrogen, incorporated with both the strands of the DNA. The DNA extracted from the second culture medium consists of what? The normal nitrogen only, light nitrogen only, and it consists of normal N14 or hybrid type only. One strand with the 15, nitrogen 15, and another one with nitrogen 14. It showed intermediate density after 20 minutes. So after 20 minutes, this, what you can say, after keeping in the heavy isotope of nitrogen, this E. coli is transferred to the normal nitrogen, and after 20 minutes, it produced the first generation and go for the centrifugation, and there they got what? An intermediate weight density. Is not light or it's not heavy. Intermediate, because one strand is heavy and another strand is what? The light only. That is what the DNA they got in the first generation. Right. Then the DNA extracted from the same culture medium consists of N14 after 40 minutes. That is for the second generation only. First generation they got after 20 minutes. They go for next division again after 20 minutes. That is 20 plus 20, 40 minutes only. After another generation, this generation composed of equal number of this hybrid DNA and light DNA. See, in first generation, both are hybrid only. But in second generation, they got equal number of what? The hybrid and what you can say, light DNA only. And that is in the second generation after 40 minutes. So same thing you can see here. This is what the diagram present in your textbook. This is what the heavy DNA, because it is formed because of the incorporation of the heavy nitrogen isotope in both the DNA strand after keeping this E. coli in that medium for many generations. And after 20 minutes, you just take this E. coli from the heavy isotope and put it into N14 medium. And after 20 minutes, normally this DNA go for replication and produce two DNA. And these two DNA in first generation are hybrid only. One is with the heavy isotope of nitrogen N15, and another one is with the light, what you can say, nitrogen N14. It is hybrid type. This is also hybrid. Why it is hybrid? At the time of what? The replication, both the strands of the DNA separated, and these both the strands are act as what? The template or the master strand, and these two strands only constructed a new one. But the new strand constructed here by using the N14 only. Why? Because it is in the light nitrogen medium only. Ammonium chloride consists of what? N14 only. That is why the second strand, it is what? The light one. And it is keep in the same medium only for getting the second generation after 40 minutes. And this will go for replication. This is also going for replication separately, independently. So it separate, and the light strand produce what? Another light strand. This will be light in a weight, and another heavy strand go for producing another replication by producing what the light strand here, because it is in the N14 only, and this will be hybrid. And same thing happened to this DNA also. So get two hybrid and two light in the same proportion, 1:1. So this is what the heavy one. Centrifugation, you see it is settled down in the, because it is heavier, both the strands are heavier. And this is what the hybrid, here only its location, the hybrid, it is not heavy, it is not light. But here they got two layer. This is what the light one, and this one is what the heavy one. After this centrifugation by using what cesium chloride centrifugation method, density centrifugation method, right? So this is what the experiment conducted by what you can say, these two scientists, and Michelson and Stachel, and here you can see the picture of this Michelson, right? Then come to the experiment conducted by the Taylor and his colleagues to prove that DNA replication is semi-conservative. And this also asked once in the board examination. It is what the qualitative proof. Whenever it is asked, the qualitative proof for what you can say semi-conservative method of DNA replication, you have to explain only the experiment conducted by the Taylor and colleagues. And this experiment is so simple. See, this is what the DNA molecule. This is what the DNA molecule here, double-stranded DNA molecule. And they label the thymine present in the DNA, with a radioactive material. They label only the thymine present in the DNA, radioactive thymine here in this DNA, and allow the DNA to go for replication, So naturally, this will be separated. One strand separate from the another. And these two strands are acting as what the templates only, and construct a new strand. And new strand, whenever it construct, thymine is not having the radioactivity present only in one strand, just like a hybrid strand we have seen there. There are two strands of DNA produced here after the replication, and both the strands are hybrid type. Hybrid type means what? Here, one is having the radioactive thymine and another one is not having the radioactivity. And newly constructed DNA is not having the radioactive thymine, whereas that old, what you can say, that master strand is having what the radioactive thymine. And such a type of experiment only conducted by the Taylor and colleagues. It is what the qualitative proof to explain the process of DNA replication is a semi-conservative one. So they conducted the experiment in a beans plant, Vizia Faba, in 1985. And this experiment also to prove that DNA replication is semi-conservative type, and it is what the qualitative proof only. And here they use radioactive thymine to label it and allow them to replicate. The thymine present in the DNA is get labeled with what the radioactive material, and allow this DNA to replicate. Only thymine, they what you can say, labeled with the radioactive material, and allow what you can say, this DNA, both the thymine present in both the strands, labeled with the radioactive material and allow them to replicate. And they found that radioactive thymine present only in one strand of DNA after replication, that is in the old strand only. That only they labeled. And newly constructed one is not having the radioactive thymine. Why? Because it is what newly constructed and it is not what you can say conserved one. So in DNA replication, during DNA replication, one strand of the DNA always conserve and another one will synthesized newly, right? And that is called as what the semi-conservative method of DNA replication, and it is what the qualitative proof for that. And they did not detect radioactive thymine in newly synthesized DNA. And whenever they observe the radioactivity of such type of thymine in the coming generation, So naturally what happened, radioactivity decrease in the coming generation, and they use autoradiography and X-ray sheet to detect the radioactivity of this thymine. Same way, you see both the strands are heavy, and whenever come to the first generation, only one strand is heavy. Come to what you can say second generation, again what happened, that heavy strand only two. Then go to the third generation, only two. The remaining DNAs are normal only. So whenever we take the DNA sample from the coming generation, there is a reduction in the what you can say number of heavy strand present in the DNA. Same thing only they have seen. The thing is that here they use what the radioactive thymine only. And that's why we call it as what the qualitative proof for what you can say this explain the DNA replication is a quality, I mean semi-conservative one. So now we will go for another topic. Who determined the height of the person? So we used to take a lot of exercise to improve the height. Of course, chance is there to increase the height 1 inch or 2 inch. But who determine really the height of the individual? Not only height is what a character. Like many characters are there in our body. Who determine the character present in the body? No doubt it is determined by what you can say the DNA present in our body only. And this DNA only express a character. That is why we consider DNA as what the genetic material, and this DNA expresses its character. How this DNA expresses its character, that only we are going to discuss now. Expression of character by a DNA by producing an appropriate enzyme in the cell, and in cellular level, this enzyme make a lot of changes that ultimately making what you can say a specific character in the individual. So normally this DNA is expressing a character only by producing a particular protein. That only we are going to discuss here in very detail. DNA as a genetic material expresses character by producing a particular protein in a cell. It may be in the form of enzyme, or some proteins are produced in the cell in the form of hormones. You know how these enzymes and hormones are working in our body, right? Then this protein can control cellular functions and also make a particular character. The protein present in the cell may be in the form of enzyme, or it may be in the form of hormones only. And by using the enzymes and hormone, individual is able to control basically the cellular functions and also make a particular character. So expression of a gene in the cell is under the control of external and internal factors. Genes we have, many genes we have in our body, and these genes are not continuously and constantly expressing. There is a time to express, and some of the genes are what you can say, only a very few genes present in our body is expressing character. And the remaining huge percentage of the genes are silent, and it expresses character only at the time of its requirement. Who make the gene to express the character? No doubt it is controlled by some factors present inside the body or some factors present outside the body. That is what the internal factor or the external factor only, right? And for example, one example mentioned here, some person, working regularly under the sunshine for a long period of time. That is what external factor is, what the sunshine, working regularly under the sunshine for a long period of time. So to resist our body from such a radiation, our body start producing the protein called as what the melanin and provide dark color. It is very much necessary to protect our body from such a heavy radiations. That melanin is nothing but a protein only. Who is producing such a protein in our body to make our body dark, to prevent such a radiations? Some genes only. So normally that gene is what you can say not expressing that demand, that what you can say some factors present in our body or some external factors only making the gene to express. Whenever you sit in the AC room, for a long period of time to do many jobs. So naturally, color will be what you can say, not that much dark. Depend on what you can say, genetic material only. But that external factors can make our gene to express the character. Otherwise, what happened normally, this will be very silent only, right? So we will see more detail about the expression of character by the DNA by producing a particular enzyme or particular hormone, right? That only we are going to discuss now in very detail. John? Yeah, that only, this only. Yeah, correct. That is good. Not this one. This same slide. Right. This one, sir? No, this slide only. Okay. Yeah. That's correct. Thank you. Okay. Let us see how a gene expresses a character. By synthesizing a proteins. We call it as what? The protein synthesis only. As I told you in the beginning, DNA is the real genetic material, can store a lot of informations. What type of information they used to store? This DNA molecule is storing the information about the nature of a particular protein produced by it. So that information only helping to produce a particular protein. So to prepare the protein by a DNA, because DNA only having that information, it is stored there. There are two steps. Maybe knowing about this two steps, one is transcription. It is the process to produce RNA from DNA. DNA is double stranded. DNA cannot use for preparing the protein directly because this protein should be produced not only one time, for many times. So we cannot use DNA directly. It is storing the information only. But this information they are passing to the RNA to prepare a particular protein. And process of formation of the DNA, RNA from the DNA is called as what? The transcription. Process to produce RNA from DNA. That means all the information present in the DNA to prepare a particular protein is just a copy to the RNA and sending RNA from the nucleus to the cytoplasm to do photosynthesis. Protein synthesis. That only we call it as what? The transcription. Process to prepare produce the RNA from the DNA is called as what? The transcription. And normally DNA produce three type of RNA to synthesize a protein to express a character in the individual. These are what you can say tRNA, transfer RNA. Whenever you write the tRNA, the T should be in a small letter only. And it is what the transfer RNA. Another one is what the rRNA. R should be small letter only, ribosomal RNA. And mRNA, M should be in small letter, is messenger RNA. So tRNA, rRNA, and mRNA. These are the three type of RNAs produced from the DNA through the process of transcription. And this transcription take place in the nucleus only because, DNA is located in the, what you can say, nucleus. There is a nuclear envelope. So naturally, no chance to produce what the RNA in a cytoplasm. It produce always in the nucleus only. I told you about a particular place in the nucleus for synthesizing RNA, and we call it as what? The nucleolus. Nucleolar region. It is what? Very dark area with what you can say denser nucleoplasm. And in that place only the process of transcription take place in the nucleus. And after producing this RNA, three type of RNA, tRNA, rRNA, and mRNA, these RNAs are taken to the cell cytoplasm from the nucleus for producing the real protein, because the information about the chemical nature of the protein is now with the RNA only, because it is just copied in the nucleus from the DNA and it is taken to the cell cytoplasm. So according to the information given in the RNA, only cell is preparing the particular process, and it is called as what? The translation. It is the process to produce the real protein, a functional protein. By using the information present in the RNA, it's just copied from the DNA. And it takes place naturally in the cell cytoplasm only, because the RNA is coming to the cell cytoplasm through the annulus or the pores present in the nucleus. And the second process, translation, naturally take place in the cytoplasm only, because many machineries are required for the protein synthesis or translation, and it is available only in the cell cytoplasm. But the machineries and enzyme required for the transcription present only in what? The nucleus. That the DNA is required, that is there in the nucleus. Then for synthesizing the RNA, this ribonucleotides are required, that is available only in the nucleus. So naturally, the process of RNA synthesis take place in the nucleus, and translation, the real process of protein synthesis take place only in the cell cytoplasm, because many machineries are required. What are the machineries required for the translation process? This rRNA is required, that is taken from the nucleus to cytoplasm. tRNA is required, that also taken to the cytoplasm. Then mRNA is required, that is also taken to the what you can say cytoplasm. Addition to that, so many amino acids are also required, because protein is nothing but the long chain of amino acids only. And these amino acids are also available only in the cell cytoplasm. That is why cytoplasm only facilitate the material for the translation process. That is what the real process of protein synthesis. See the central dogma of eukaryotic cell, and even what you can say prokaryotic cell, and in case of what you can say this individual like virus having the RNA as a genetic material, they go for the reverse transcription in the central dogma. Reverse transcription, because they don't have the DNA, double-stranded DNA. Their genetic material is RNA only. Retroviruses, we discussed in the morning. Retroviruses or having only RNA, and this RNA go for reverse transcription. Transcription is what? The process to produce RNA from the DNA only. We are all what you can say having the DNA as a genetic material. So normally DNA go for transcription and produce RNA. Every information present in the DNA for synthesizing a particular protein is just a copy during the transcription in RNA. And by using, after the production of RNA in the nucleus, it is taken to the cytoplasm. And by using the machinery, this RNA used for preparing a particular protein, and that will be called as what? The translation process. At the same time, DNA can replicate. It can multiply. Before every cell division, what happened? It go for replication. We discussed about the semi-conservative method of DNA replication that is happening here in the nucleus. Whereas in case of what the retroviruses, they have only what? This RNA as a genetic material. So what happened? By using RNA, it go for reverse transcription, means production of DNA from this RNA. Then go for transcription, produce RNA. By using RNA, go for protein synthesis in case of what? The retrovirus. These retroviruses having only mRNA as what? The genetic material. And this is what the two events happening during the process of what? The protein synthesis. That only we call it as what? The central dogma. DNA to RNA, then RNA to proteins. The DNA to RNA, then RNA to protein. DNA information present in the DNA is just a copy through the process of transcription in RNA. Then it's go for translation, and by using the information copied from the DNA, it is preparing a meaningful protein, and that only we call it as what? The translation. So let us move to the first event in this central dogma. That is what? The transcription. So tell me, what is transcription? What is transcription? It is the process to produce what? RNA from the DNA. What is the need to produce RNA from the DNA? Because all the information required for synthesizing a particular protein is already there in the DNA in the form of nitrogenous base pairs. That sequence of nitrogenous base pair in the DNA only determines the chemical nature of the particular protein synthesized. And this information just copy to the RNA through the process of transcription, because the cell cannot utilize the DNA directly for the synthesis of protein, because protein synthesis in the cell cytoplasm only. So we cannot take, the cell cannot take this what? The DNA to the cytoplasm. So that is why it is producing a copy. What are the, because DNA is what? Very lengthy one. Cannot, it is not possible to take the entire DNA, but for preparing a particular protein, a small information only required, not the entire DNA. A small area of the DNA only acting as what? One cistron to produce a particular protein. So only that part to be copied, not the entire length of the DNA. No need to copy the entire length of the DNA. Which part of the DNA is responsible for a protein, a specific protein, and that part of the information from the DNA only just copied to the RNA, and that only taken to the cytoplasm for translation. So this process of copying genetic information from one strand of the DNA to RNA. One strand of the DNA only used here. We will see while discussing about the transcription. Actually, DNA is double-stranded. But to synthesize RNA, we required only one strand. That only acting as what? The template strand or the master strand to produce new RNA. DNA help for constructing RNA strand is called as what? The master strand. I told you only one strand is required for synthesizing RNA from, it is exactly like what? The DNA replication only. You know, in DNA replication, making new DNA strand by considering one strand as a master strand. But in replication, both the strands are master strand only. That is why they are producing two new strands. But in case of transcription, remember, only one strand is acting as what? The template strand. Other is not template strand. It is what? The coding strand only, not at all involving in the process of what? The transcription. And because of that, only one strand will be produced during the transcription. And this strand is not DNA, because in DNA replication, only DNA polymerase enzyme is used. But here, another enzyme is used for the elongation of this RNA. And we call it as what? The RNA polymerase. It is DNA-dependent RNA polymerase. It is to make a new strand of RNA polymerase, but it elongate in the same direction as what the DNA polymerase elongate. Remember, it's always elongate from the 5' direction to the 3' direction only. So DNA help for constructing the RNA strand is called as what? The master strand. It is like a DNA replication only. But here RNA is synthesized by using DNA-dependent RNA polymerase enzyme, not the DNA polymerase enzyme. So DNA polymerase enzyme required a primer to attach, that we have seen in case of what? The DNA replication. Whereas for preparing this RNA, that primer is not at all required. So while discussing the transcription and transcription and what you can say replication, you just compare it. Lot of similarities are there, at the same time so many differences are also there. You have to understand that and develop the concepts. And here this elongate the new RNA strand in the direction same 5' to 3' direction only. DNA-dependent RNA polymerase plays only the uracil instead of thymine. That you have to remember. DNA polymerase enzyme only elongate the DNA. In DNA, that thymine is what? One of the nitrogenous base pair that is not there in RNA, because it is what? The RNA synthesis only. RNA polymerase only is used here to elongate the new strand of RNA by considering one DNA strand as what? The template. So naturally, instead of thymine, this RNA polymerase enzyme plays only the uracil, and this process takes place in the nucleolus within the nucleus. You know, the DNA, from the DNA only just it's copied to the RNA, and DNA is located inside the nucleus, it never comes out. So naturally, the process of the transcription takes place only in the nucleus, particularly in the nucleolar region. See, this is what the DNA. So actually, the entire length of the DNA is not required. There is some specific area in the DNA to produce a particular protein. Entire length of DNA is not for one protein. For producing a particular protein, informations are available in the DNA only in a particular place, not the entire length. So in which area it is located, that area of the DNA only used for preparing this new RNA. This information present in the DNA just copy to the RNA only. And there are two strands in DNA. Here you can see there are two strands, and just like DNA replication, for producing RNA in between, that hydrogen bond should be broken, and it is broken with what you can say helicase enzyme only, just like what the DNA replication. After breaking it, naturally these two strands will be separated. And consider one strand as a template, another one is not the template, it is coding strand only. So naturally what happened, based on the nitrogenous base in the template, that new RNA will be elongated by the RNA polymerase enzyme from 5' direction to 3' direction. For the elongation of RNA to bind this RNA polymerase enzyme, that what you can say primer is not at all required. Primer is not required. So it can elongate like this in the direction 5' to 3' direction. So from here it elongate. Whenever it's what you can say elongate, naturally what happened, this will move like this, and these are all what you can say broken only, and later after taking this RNA out, again it go for recoiling and getting its what exact shape. Whenever this process of transcription complete, then what happened, the DNA go for coiling, and it is getting what the three-dimensional original shape, because it has to use for producing the same protein in the next time. So it should be there as a, what you can say, three-dimensional structure permanently in the cell. That is why it is considered as what the what you can say storage gallery for information related to a particular proteins. And this process only we call it as what? The transcription. So as I told you, the entire copy of the DNA strand is not completely used for the transcription. Which area is having that particular information for a protein, a functional protein, that area only should be copied into the RNA. The region of the DNA coded for a particular protein only, undergo the process of transcription, only one strand is used to copy RNA in transcription. What will happen? Both the strands of the DNA is used for copying RNA. Both the strands of the DNA suppose produce RNA. What will happen? Both the strands of DNAs are complementary. All of you know adenine, thymine, guanine, cytosine, like a combination we can find in the DNA. Both the strands are antiparallel and it's complementary. Nitrogenous bases are what? The complementary pairs only. Suppose both the strands are producing RNA. Two RNA will be produced and both RNAs are also complementary because it is copied directly from the same place only. Some place of the DNA only. So naturally the RNA also will be complementary. So it will go for pairing and produce what? dsRNA, double-stranded RNA, instead of what? The DNA, double-stranded RNA will be produced. Actually, one strand of RNA is required for synthesizing protein. If it go for producing double-stranded RNA, that process become more and more complicated. So to avoid such type of complication during the transcription or translation, only one strand of the DNA only acting as what? The template, and by using this template, it produces only one strand of RNA. So only one strand of DNA is used for copying the RNA in transcription. If both the strands are used for transcription, it produces two RNA of two different type, and these RNAs are complementary and having what? The same kinds of sequence of nitrogenous bases. So it go for pairing and go for producing double-stranded RNA, dsRNA or double-stranded RNA. So naturally, that will complicate the process of protein synthesis because protein synthesis required an information just copied from the DNA. Because in protein, during protein synthesis, the cell should understand what type of amino acid will be placed one after another. That information only given by the DNA in the form of RNA. Only one strand is required. If both the strands of the DNA is going for the transcription, naturally it produces two RNA, and these two RNAs are complementary, and these complementary strands go for pairing and produce double-stranded RNA. It makes the what you can say, this process so complicated. So come to the types of RNAs transcribed. So as I told you, there are three types of RNA. There are three types of RNA transcribed from the DNA. One is ribosomal RNA, rRNA. Second one is what? The messenger RNA, mRNA. And third one is what? The transfer RNA or tRNA. And to synthesize these three different type of RNA, three different type of polymerase enzyme also used. RNA polymerase. RNA polymerase means what? The enzyme used for the elongation of an RNA strand from the DNA by using the DNA as a template. And to produce what? The ribosomal RNA, RNA polymerase 1 only used. RNA polymerase 1 for synthesizing the rRNA. RNA polymerase 2 is used for synthesizing what? The messenger RNA or mRNA. Then RNA polymerase 3 is used for synthesizing tRNA. To remember this, you have to make a code like this: RMT, rRNA 1, number 1 polymerase, M, messenger RNA, that polymerase number 2, and tRNA, polymerase number 3. So there are three types of RNA polymerase in the nucleus. And RNA polymerase 1, it is to synthesize what? The rRNA from the DNA through the process of transcription. And messenger RNA or mRNA is produced in the nucleus by using that RNA polymerase only from the DNA. And transfer RNA, tRNA is synthesized from the DNA by using another enzyme called as what? The RNA polymerase 3. But always, all these RNA polymerase enzymes are elongating from the 5' direction to 3' direction only by considering only one strand of DNA as a template. See, actually, this rRNA is common for all kinds of enzyme production, all kinds of protein synthesis. It is common for all type of enzymes. And tRNA is also common for all type of enzymes. If you want to make any type of protein in the cell, we can use rRNA because only one type. And tRNA also like that, only it's common for synthesizing any protein in the cell. Whereas this what the mRNA, it is varies from protein to protein, because this sequence of nitrogenous base present in the mRNA only determine what type of amino acid will be placed one after another during the process of protein synthesis. And this is not common. It is depend on the nature of the protein. Nature means what? The total number of amino acid present in the protein or length of the protein, polypeptide chain. It is determined by the mRNA only, not by the rRNA or not by the tRNA. And these tRNA and rRNAs are common for all type of protein synthesis in the cell cytoplasm. But this mRNA is very specific, and that only determine the nature of the protein synthesized. And that only determine total number of amino acid present in the protein or the length of what the protein synthesized in the cell. Right? So there are three types of RNA production in the nucleus through the process of transcription by using three different type of DNA-dependent RNA polymerase. Why it is called as DNA-dependent? Because it is what the DNA, it is what the single DNA strand, and new strands of DNA, it is what the 3' end and 5' end. And this strand having 3' and 5' end only acting as a template, because RNA is synthesized here from the 5' direction to 3' direction only. So it should be 3', 5', and that only what the what you can say master strand or the template strand. And another strand of DNA is called as what the coding strands only. Right? And transfer RNA and ribosomal RNAs are common for all type of protein. But the chemical nature of the messenger RNA vary from protein to protein. That only determine the chemical nature of the protein synthesized. That only determine the total number of amino acid present in the protein. That only determine the sequence of amino acid present in a protein or polypeptide chain. Then let us see the transcriptional units. See, this is what you can say unit for producing a particular mRNA or RNA, that is called as what the transcriptional unit. It is what the DNA only, the DNA strand only. One is running from the 5' direction to 3' direction, and another one you can see 3', 5' direction only. This is what the DNA strand, and this DNA strand only acting as what one unit. And this only transcribe a particular RNA having a meaning, produce what? A functional enzyme or functional protein. And in one unit, this unit produce what? RNA. And this unit is called as what? The transcriptional unit, consists of mainly three areas. One is what? The promoter. And here we can find the terminator. And in between the promoter and terminator, there is what? Sequence of nitrogenous base in the DNA, and it is called as what? The structural genes. So three components in a transcriptional unit: promoter, then a structural gene, and terminator. See, promoter is to start, and the terminator is to stop the process of what? The RNA synthesis. And the chemical nature of the RNA. Chemical nature means what? The nitrogenous base sequence present in the RNA produced from the transcriptional unit. It depends on what you can say nitrogenous base present in the structural gene only. And here, which gene, which one is acting as a template? that what you can say 3' 5' strand only acting as a template, because the promoter only initiate the new RNA. From here only new RNA will be synthesized. So here the synthesis of RNA from the promoter always from the 5' 3' direction only. Why? Because RNA polymerase always elongate a new strand of RNA from 5' direction to 3' direction only. So naturally that 3' 5' strand of the DNA only acting as what the template, because this newly constructed RNA is always complementary to the template strand. Always it's complementary to the template strand. Suppose here adenine is there, or suppose here guanine is there. In case of RNA, here uracil will be there because thymine is not there, and here guanine. So naturally cytosine will be there. So the nitrogenous base sequence in this RNA is depend on the nitrogenous base sequence present in the template strand only. And another strand of this transcriptional unit of the DNA is called as what? The coding strand. Actually, coding strand is not having any role to determine the chemical nature of what the RNA synthesized in between. But this newly constructed RNA in between these two strands, by considering this strand as a template, sequence is exactly like what the coding strand only, because these two strands are complementary. For example, here A is there, naturally here T will be there. Right here I told you, right here you can see that A, here what you can say, here G, here C, and actually here also C only, G, C only, and here it is A, and here it will be T. So in case of what the DNA, T will be there in coding strand. Wherever a T is there in the coding strand, in case of RNA, instead of T, what will be there? U will be there. That is what the only difference in the sequence of nitrogenous base in the RNA. I think you are getting the idea correctly, right? So there are 3 units in the transcriptional unit. One is promoter, that is to initiate, and there is a factor present in the promoter that is called as what? The sigma factor, that only helping to initiate the process of what? The transcription. And it is called as what? The promoter. And here there is a factor that is called as what? The raw factor here. And this raw factor present in the terminator, that only terminate the process of what? The transcription. So whenever this RNA elongate and reach to the terminator, that RNA polymerase enzyme detach and stop the process of what? The elongation and process of transcription. And sigma factor only help to attach the polymerase enzyme, RNA polymerase enzyme. So that is why it's initiate or start from this promoter region, and it is stopped in the terminator region. And the chemical nature of the RNA produced in between the promoter and terminator exactly like what the coding strand, except what? The thymine. Instead of thymine, uracil will be there, because the template, is here it is working as a template. Here the 3' 5' strand only. And one more thing, that 5', I mean that promoter is always located in the upstream position, upstream, and the terminator in the downstream. Why it's called as upstream? And see the coding strand, that promoter is located in the 5' end of the coding strand. And not only coding strand, that promoter is located in the 5' direction of the newly constructed RNA. And that is why this end is called as what? The upstream. Whereas this terminator is located in the 3' direction of what? The coding strand, and also located in what? The 3' direction. This is what? The 3' direction of what? The newly constructed RNA. And it is exactly like what? The coding strand only in structure. As I told you, it's RNA only, so that there will be no thymine, only uracil. And it is what? Pentose sugar is not deoxyribose sugar. This will be what? The ribose sugar only. Right. So that you have to apply, that the things that we learned earlier should be applied here while constructing the RNA in between what these two template strand as well as the coding strand. Template strand only used for constructing new strand of RNA should be complementary. And so this area is called as what, the structural gene. Why it is called a structural gene? Because the structure of the newly constructed RNA is totally based on the nitrogenous base present in this area between the promoter and the terminator only. Raw factor is there in the terminator that never permit the RNA polymerase to extend further. And it is located in the 3' direction of the coding strand and also located in the 3' end of what newly constructed RNA here. That is why we call it as what, the downstream. It is called as what, the downstream, and this one is called as what, the upstream. See the arrow, this arrow indicating what, the elongation of the new strand of DNA in between these two from the promoter to the terminator only. And this unit only we call it as what, the transcriptional unit, right? Here we can see that transcriptional unit. Another diagram is here. This is what the length of the DNA having two strands here. You can see it is a double-stranded one. One is running from 5' strand to the 3' strand, and another one is what, the 3' strand to the 5' strand. And here we can find the promoter that is in the upstream position of what, this coding strand or the newly constructed RNA, upstream only. And this is what, the downstream where this terminator is located. And there are two strands of DNA. This one is 5' 3' and another one is 3' 5'. You tell me which strand is acting as a template strand here or the master strand. Is it 3' 5' or 5' 3'? It is starting from the promoter only. Okay, it is always elongate from the 5' new RNA elongate always from the 5' to 3' only. So naturally 3' 5' strand only act as what, the template strand. This only act as what, the template strand. And this is what, the non-template strand only, or we call it as what, the coding strand. Non-template strand or the coding strand. And this is what, the promoter, and this is what, the terminator, and this is what, the structural gene between the promoter and the terminator. The chemical nature of the RNA synthesized here in between these two strands depend on what you can say, nitrogenous base sequence in the that template strand only. This is what the template strand. Suppose here it is what, a Z, here G will be there. And if it is what, the T, here it is T, naturally what, here A will be there. And if it is what, the C, naturally G will be here. And if it is what, the A, and suppose here it is what, the A, and here U will be there. So like that only new strands will be elongated from the 5' direction to the 3' direction. This is what the 5' because template here it is complementary. So naturally this 3' and this is what, the 3' and only this is 3' and it should be 5' because it is complementary and anti-parallel. This is what the way through which cell produce a new RNA in between these two DNA strands. And it is just to copy the information present in the DNA to the RNA. And that RNA only used for protein synthesis. Then see the details of the transcriptional unit. See the transcriptional unit, as I told you, consists of the three regions. Promoter, it is to initiate the process of transcription. Then structural gene, it is to elongate the RNA strand from 5' to 3' direction by using what, the RNA polymerase enzyme. And terminator, it is to stop the process of the transcription. And one transcriptional unit is a piece of double-stranded DNA only, and it helps to produce what, a meaningful RNA. Meaningful RNA means what? This RNA synthesizes a protein having a function in the cell. That is what the meaningful RNA. This RNA is having a meaning. because it translate and produce a protein, and this protein produced by this RNA is having a definite function in the cell. Either it work as a hormone or it work in the cell as what the enzyme for a definite function to express a character. See, double-stranded DNA consists of promoter to initiate the process of transcription, as I told you, and the transcription unit end with the terminator, that is to terminate, to stop the process of transcription, and also consists of a structural gene between the promoter and terminator, and that only decide the real chemical nature of RNA synthesized newly. The chemical nature. Chemical nature means what? Nitrogenous base sequence. That is what the chemical nature of the RNA. The arrangement of what? The ribonucleotides. That sequence only determine the meaning of the RNA synthesis, and it is depend on what? The structural gene present in between the promoter and terminator only. This is what the structural gene between the promoter and terminator, and this is what the promoter to initiate, and this is what the terminator to stop the process of transcription. Here we can find a factor called a sigma, and here we can find the rho factor, and the sigma factor only attach the RNA polymerase, DNA dependent RNA polymerase, and it elongate. Whenever it touch the rho factor, it stop the further elongation. With that, what happened? This will be detached. And during this transcription, what happened? Whatever the area transcribed, that will come out from what you can say DNA, and this part of the DNA go for coiling, like that it will coiling, and this will elongate. According to that, it will go for coiling. After completing what the RNA synthesis, reaching here in the terminator, RNA synthesis will be stopped. So it is getting back, the DNA is getting back it's what the three-dimensional coiled structure. So information is completely stored in the DNA. Whenever cell want to produce the same protein later, half an hour or after 2 days or after some years, so this information is ready all the time in the DNA to express the same proteins. So that is why these DNAs are not taken from the nucleus to synthesize the protein. And the DNA is always storing the information for a character. Permanently, it is there in the chromosomes or in the DNA, right? And this only we are going to discuss here in detail. And the first one is promoter. What is the role of the promoter in transcription? It is to just initiate the process of transcription by considering one strand as a master strand. Which strand of the DNA is acting as a master strand? That is a 3' 5' strand only. That is what the what you can say the master strand or template strand, and the other strand having 5' 3' strand is what the coding strand only, not having any role to code the new RNA synthesized in between. But the newly synthesized RNA is exactly like what the coding strand of the DNA only in the sequence of nitrogenous bases, except what the thymine. No thymine in RNA. Instead, uracil will be there. So come to the details of the promoter. It is what the sequence of nucleotide present in the transcriptional unit only to initiate the process of transcription. There is a factor present in the promoter. To initiate the process of transcription, it is called as what? The sigma factor or initiation factor. What is the role of the initiation factor? That sigma factor or initiation factor present in the promoter region help RNA polymerase to initiate the process of transcription. I told you 3 types of RNA polymerase to synthesize rRNA, tRNA, and mRNA, right? The polymerase 1, 2, and polymerase 3, and all these DNA-dependent RNA polymerase attached to the sigma factor only to start the process of what? Transcription, and is located in the promoter region, and promoter region is located in the upstream. Then promoter is located towards the 5' end that is what the upstream end of the newly constructed RNA, or it is there in the 5' end of the coding strand, not template strand, that is in the coding strand. Only in case of template strand, it is in the 3' direction. Whereas in coding strand, it is in the 5' direction, and it is also in the 5' direction of what the newly constructed RNA. Then come to the terminator. As I told you, this process of what the transcription get terminated when this RNA polymerase reach to the terminator, because there is a factor here called as what the Rho factor. It is a sequence of the nucleotide present in the transcriptional unit at the end only. And this is the sequence of nitrogenous base to stop the process of transcription. That's why called as what the terminator. There is a factor present in the terminator to stop the process of transcription, and it is called as what the Rho factor that only terminate the process of transcription, and termination is located at the 3' end of the newly constructed RNA, and it is also located in the 3' end of the coding strand. So we call it as what the downstream end. Upstream and downstream end, and that promoter is there in the upstream end of the DNA, whereas the terminator is in the downstream only. And in between this, what you can say, promoter and terminator, there are many sequence of nitrogenous base in the template strand or the coding strand, and it is called as what the structural gene. Why it is called as structural gene? Because this area only determines the chemical nature of the RNA synthesized newly. That is why we call it as what the structural gene. And promoter is only to start. Terminator is only to stop. But this RNA synthesized in between the DNA depend on the nitrogenous base sequence present in between the promoter and terminator, and that only we call it as what the structural genes. And this is what the double-stranded DNA segment present in between the promoter and terminator. Here, this is what the double-stranded DNA only between what you can say the promoter and the terminator. And this sequence of nucleotide present in the area only decide the chemical nature of the RNA synthesized newly. And this DNA strand consists of two strands. One is 3' 5' strand, and another one is 5' 3' strand. And the 3' 5' strand of the DNA in the structural gene only acting as a template. Why? Because RNA, newly constructed RNA is always elongate in the direction of 5' 3'. So it should be complementary to the DNA strand, template strand. So naturally that 5' 3' strand of the DNA only acting as a template for a new RNA strand, or it is called as what the master strand, or we call it as what the template strand. Whereas 5' 3' strand, another strand of the DNA in the structural gene, it stands of structural gene only, and it's called as what the coding strand, and it never code for any RNA. But the chemical nature of the RNA synthesized newly in the structural gene is exactly like what the coding strand only. Only one difference in coding strand, thymine will be there, but in case of what the RNA instead, uracil will be there. And another one, this what the pentose sugar present in this what the coding strand is what deoxyribose sugar, because it is a part of the DNA. Whereas the pentose sugar present in the newly constructed RNA is what the ribose sugar only, right? And newly, what you can say, RNA is being synthesized and elongated by DNA-dependent RNA polymerase enzyme in the direction always 5' 3'. Remember the DNA-dependent DNA polymerase in case of what the what you can say replication. DNA-dependent DNA polymerase only used for the what you can say DNA replication, and that also elongate in the direction 5' 3' only. That is why we have seen what one leading strand and another one is what the lagging strand. In lagging strand, that new DNA is constructed in the form of what Okazaki fragment. Why? This permit the DNA-dependent DNA polymerase always permit the elongation of the DNA in the same direction 5' 3' only. That is why it is produced in the form of fragments there. And RNA polymerase also elongate the new RNA from 5' to 3' direction only. So gene is a sequence of DNA nucleotide code for a particular RNA to produce a functional protein. What is gene? Nothing. Chemically, it is what the DNA only, a particular length of the DNA, and this DNA is coding for a particular RNA, and this RNA is functional and produce what a protein with a definite function. That is what the gene. Maybe heard about this term. This gene is chemically it's the DNA only, a particular length of the DNA, and this DNA is having what the transcriptional unit, and it synthesizes RNA. And this RNA is meaningful and produce what a functional protein in the cell for a particular character. And this sequence of DNA nucleotide code for a protein or a polypeptide chain, and it is called as what the cistron. A sequence of DNA nucleotide. It is what the sequence of DNA nucleotide, and it is called for a particular mRNA or RNA, and this RNA produce what a particular protein. It produce what a particular protein, right? And this is called as what the cistron. This is what the sequence of nitrogenous base present in the DNA for a particular polypeptide chain. This RNA is called for a particular polypeptide chain, and we call it as what the cistron. And sometimes one structural gene code for one polypeptide chain. One structural gene, it is coding only for what one polypeptide, and it is producing an RNA, and this RNA produce only one polypeptide chain. And such a type of cistron is called as what the monocistronic gene. It is what the gene produce what one polypeptide chain, because this gene transcribe a RNA, and this RNA produce only one polypeptide chain, and we call it as what the monocistronic. The gene is monocistronic. And most of the structural genes in eukaryotes are monocistronic only. If a structural gene of what a gene is coding for more than one polypeptide chain, means this is what the structural gene, it is producing an RNA, and this RNA is for more than one polypeptide, and we call it as what the polycistronic genes only. Monocistronic gene and polycistronic genes. Monocistronic genes are the structural gene produce a RNA, and this RNA is for a single polypeptide chain or a protein. That is monocistronic only. Sometimes in case of what some individual, we can find some structural gene produce an RNA, and this RNA is not only for one protein, for many protein, more than one protein, or the polypeptide chain, and that only we call it as for the polycistronic. And most of the structural genes in the prokaryotes are polycistronic only. Whenever you discuss about the prokaryotes and eukaryotes, because in examination they used to ask such type of questions, you should have thorough knowledge about what the things happening in the prokaryotes and eukaryotes during what the transcription, translation, then DNA replication, etc. That is why we are discussing and all these things in very detail. So chances are there to ask. Many times it's asked also. So most of the structural gene in the prokaryotes are polycistronic only. Polycistronic, whereas in case of eukaryotes, most of the structural genes are for one polypeptide or for one proteins. Right. Come to the process of transcription. Process of transcription. By using the transcriptional unit only, DNA produce what? A new RNA inside the nucleus. There are 3 steps. 3 units are there. One is what the promoter, that is to initiate the process of this RNA synthesis. Then structural gene, there we can find the increasing the length of this RNA, that is called as what, the elongation. And another area, that is what, the terminator region. Whenever this elongated RNA comes in the terminator region, because of the Rho factor, what, the process of what, elongation will be stopped. And that only we call it as what, the termination. Initiation in the promoter region, elongation in the structural gene, and termination in what you can say, terminator region. And these are the three processes involved in the process of transcription. See the detail of what this transcription, right? It is what, the promoter. There we can find the sigma factor. And the RNA polymerase enzyme, three types are depend on what the type of RNA being synthesized, tRNA, rRNA, and mRNA. And RNA polymerase attached to the sigma factor. Then this RNA polymerase plays the nitrogenous bases, that is what, the nucleotide only, ribonucleotide only, because it is what, RNA only is being synthesized here. So ribonucleotide will be placed according to the sequence of nitrogenous base present in the template. It will be placed, it should be anti-parallel and placed one after another, and it will elongate from the 5' direction to 3' direction. This what the RNA polymerase. And whenever it elongates, naturally this new RNA strand also produce, and it will come out, and this will go for the coiling again and regain its what, the real structure. And this Rho factor is taking to what you can say, the terminator region. Once it come to the terminator region, Rho factor is there. That Rho factor, what you can say, attach the polymerase, then it is detached from this structural gene, and with this the process of that elongation will be stopped. That is called as what, the termination. Simple step, simple process, right? Initiation by using the sigma factor present in the promoter region. Then after attaching the RNA polymerase in the sigma factor, naturally it elongates from the 5' 3' direction by considering the template. Which is the template? 3', 5' stand only considered as what, the template. This stand only considered as what, the template. And this stand is what, the coding stand only. So it will be placed all these what you can say, the nucleotide, ribonucleotide, one after another, according to the nitrogenous base sequence present in the template stand, and it go for elongation, and RNA polymerase only will elongate the process, and it will go, and finally it reach to the terminator region. Their Rho factor is there, it attached to the Rho factor, and finally what happened, this will fall, and with that the process of RNA synthesis will be stopped. So RNA will come out, and DNA, it is regaining what, the real structure, and store the information, and it will be in the nucleus for producing the same kinds of what, the protein later. And this is what the process of what, the transcription. Initiation is there, elongation is there, and termination also there, right? And come to the initiation process. The same thing only mentioned here. Initiation, DNA-dependent RNA polymerase. Why it is called as DNA-dependent? Because here RNA polymerase is using the DNA as a template. The DNA-dependent RNA polymerase enzyme bind to the sigma factor present in the promoter located in the upstream, that 5' end of the coding strand only. Then hydrogen bond connected to the two strand of the DNA. DNA is what, the double stranded only, and it is what, the upstream position, and it is connected each other with the hydrogen bond. So what happened? The hydrogen bond connected these two strands of the DNA, broken down by an enzyme. Already we discussed this enzyme, and it is called as what, the helicase enzyme only. After breaking this hydrogen bond, what happened? It separates this DNA strand, and acting as that 5' 3' 5' strand only acting as a template, and other strand, 5' 3' strand is what, the coding strand only, and no need of primer here to start the RNA synthesis, because for the DNA synthesis only primer is required. Primer is nothing but the RNA primer, a small strand of RNA, and from there only that new strand of DNA produced in case of what the DNA replication. But here RNA, to synthesize RNA, primer is not at all required because it's already what RNA only, right? And here we will see another one, that is what the elongation. See, after initiation, here for elongation. The DNA-dependent RNA polymerase enzyme, as I told you, it's elongate the RNA by using 3' 5' DNA as a template. This is what the two DNA strand here, two DNA strand, and it is 3' 5' strand only act as a template here. Another one is coding strand only. And for the synthesis of these three different type of RNA, tRNA, rRNA, and mRNA, required three type of RNA polymerase enzyme. Which are the three type of RNA polymerase enzyme? RNA polymerase number 1, 2, and 3, right? And enzyme in eukaryote. In prokaryote, single RNA polymerase can prepare all type of RNAs. Remember, keep it in your mind. Not like what the eukaryote, prokaryotes, like what bacteria, single RNA polymerase, no need of three type of RNA polymerase. Single RNA polymerase is able to produce all the three type of RNAs, tRNA, rRNA, and mRNA. And RNA polymerase enzyme will place ribonucleotide because the synthesizing RNA only. So ribonucleotide only place ribonucleoside triphosphate will be placed one by one by this RNA polymerase according to the nucleotide sequence present in the template strand, because it should be complementary to the template strand only. And RNA polymerase enzyme elongate only in the direction 5' to 3', and only a short stretch of RNA bound to the RNA polymerase enzyme. It go for elongation from the 5' direction to 3' direction. That is only in the structural gene area. And like that, it will elongate. Now it reaches to the terminator region where the process of what the transcription will be terminated or it will be stopped. So the next process, that elongation, that we will discuss tomorrow in the morning. So be with me at 6:30 in the morning. So we will discuss the last part of the transcription, that is termination. Then we will see the real process of protein synthesis that is happening in the cell cytoplasm by using all the machinery, tRNA, rRNA, mRNA, and that amino acid present in the cytoplasm. So that we will discuss tomorrow. So take care, and we will meet tomorrow morning at 6:30. So good night to all of you, and go for your dinner. Thank you so much. Into the match. 60 was done. 7. Who got this much?